Assalamualaikum and a very good day to everyone. My name is Fatin Rasha binti Ismail. I am a sales representative of Jasin Biotech Sendirian Berhad. Technologies for plant transformation are important for advanced molecular plant genetics and crop enhancement through genetic engineering. Since the 1980s, binary vectors have been designed to contain a broad variety of selectable markers and reporter genes for agrobacterium-mediated transformation. Agrobacterium tumefacient-mediated processes are widely used to achieve stable plant transformation. One of the most widely used binary vectors in the agrobacterium-mediated transformation of plant is PGREEN. Thus, I would like to present to you our versatile and flexible binary vector for agrobacterium-mediated plant transformation, which is PGREEN binary vector. So, this is the contents of what I will be presenting today, which includes 4 parts as shown in here. PGREEN binary vector is a series of binary vectors that configured for ease of use and to meet the demands of a wide range of transformation procedures for many plant species. It first described in 2000 as component of a novel tDNA binary system and remains one of the most widely used for plant transformation until now. This binary vector able to clone specific pieces of DNA from one plant and transfer them into the genome of a different plant. This produces a genetically modified plant in a process known as agrobacterium-mediated plant transformation. The binary vector PGREEN has special features that have been addressed for the improvement of the binary vector. PGREEN able to replicate in E. coli, however, it is unable to replicate in agrobacterium without the presence of another binary plasmid, which is PSOUP, in the same strain. PSOUP being the co-resident of the PGREEN and provide replication function in agrobacterium. But firstly, let me explain what is the element of this binary vector. Basic PGREEN binary vector, as you can see in this figure, consists of 2 main regions, which are the tDNA region and the vector backbone region. The tDNA region is flanked by the left and right border regions and includes the multiple cloning sites based on the P-blue script vector, which allow any arrangement of the selectable marker and reporter genes. And next is the vector backbone region, containing the functional elements that enable replication in E. coli, which is the PSA origin and called E1 origin. This backbone region also contains bacterial selectable marker, which is the NPT1 gene that is encoding the kanamycin resistance. Because this PGREEN is unable to replicate in agrobacterium, it is maintained in agrobacterium by replicates provided by PSOUP, the helper plasmid. PSOUP contain the functional elements that enable replication in agrobacterium, which is the PSA replicase gene, the REP-A, and also contain the bacterial selection marker, which is the TET-R gene that confers tetracycline resistant, hence the plasmid can be maintained in agrobacterium. The popularity of the PGREEN binary vector system is in part thanks to its versatility, which means the special features of this binary vector. The special features number 1 is the small size of the vector. A basic binary vector of less than 5.5 kb were produced by the expression of the ORI and REP-A gene of the PSA locus of replication in agrobacterium. The small size of the molecules make it easy to clone and transform into and out of bacteria. This involves the origin of PUC replication, which makes the high copy number replication in E. coli, hence allow the insertion of the multiple genes can be transferred. Next, this binary vector has a modular construction that allows flexible transformation selection, where it allows for easy replacement of the promoter and terminator. PGREEN also has a great multiple cloning site with 6 BP palindromic unique restriction enzyme sites that allow full flexibility with restriction-based cloning, either for ligation or to prepare generic low-cost Gibson fragment. And lastly, it is cost-effective and has a less time-consuming when using it. Since its development, PGREEN has gone global and today used in thousands of labs on a range of different projects. And I will show you the scientific studies that use this binary vector for the plant transformation. Firstly, for the wet transformation. Over the past 20 years, PGREEN has kept up and evolved with modern cloning techniques. For example, when gateway cloning, which is a way of recombining genes into the vector, became popular, many gateway-compatible PGREENs were developed. PGREEN binary vectors have been produced that are golden gate clonable, such as the P golden green gate (PGGG). The construction of the plasmid has been shown in Figure 2. Why we chose to develop this PGREEN-based vector for the wet transformation is because there is evidence that additional agrobacterium virulence genes can increase transformation efficiency. These additional virulence genes can be expressed on PSOUP within the agrobacterium. As a result, in Figure 3, we can see this binary vector shows the higher transformation efficiencies compared to other binary vector. And not just that, another studies that using this binary vector is the Canal Transformation studies. Canal is an economic and ecological fiber crop but suffers severe losses in fiber yield and quality under the stressful conditions of excess salinity and drought. Therefore, in order to obtain new cultivars of Canal that could face and overcome abiotic stress, it is crucial to have a suitable protocol of genetic transformation. Thus, study were carried out on the Canal Transformation using agrobacterium tumefaciens. The agrobacterium strain harboring the PGREEN binary vector that carries the MPT2 gene for selection and VVWRKY2 gene were used for transformation. Construction of the plasmid is shown in Figure 4 and the presence of the transgene and its stable expression were confirmed by PCR that has been shown in Figure 5. This has been shown that this binary vector was efficient and fast, by which stable transgenic flowering plants were obtained within a short period of 3 months with 6% of transformation efficiency. In conclusion, using PGREEN binary vector are demonstrated to be feasible in agrobacterium-mediated plant transformation technology. This can be achieved by either with the PGREEN or PSOUP vector carrying the target interest gene. This binary vector were clearly not independent, but the system is unique, that the PGREEN depends on the PSOUP to replicate, thus the possibility of the co-transformation is increased. And this is the list of references of my presentation, and that's mean we have come to the end of my presentation. Make your choice wisely, and thank you so much for listening to my presentation.